Hello, everyone. Welcome to this webinar as part of our 2021 Analytical Extravaganza. I'm Brian Field, LCMS Sales Specialist for the Pacific Northwest, Rocky Mountain States, and Alaska at Shimadzu Scientific Instruments, and I'll be your moderator today. We will hear from Professor Richard Van Bremen, Principal Investigator at Linus Pauling Institute and Professor of Medicinal Chemistry in the Department of Pharmaceutical Sciences at Oregon State University, about affinity selection mass spectrometry for the discovery of pharmacologically active natural products. Before we start, I wanted to share a couple of notes for our viewers. The webinar console has a variety of items to help enhance your experience and interaction with us. In the screen, you'll see the following items. The slides will appear on the left-hand side. Directly under the slides, you will see a resource list with clickable links relevant to the material being presented today. On the top middle is the widget for questions and answers. Please submit your questions during the presentation through this widget, and we will answer them during the Q&A session at the end. Just below the Q&A box are the moderator and speaker bios. You may expand the items here to learn more about us. On the right are survey questions that you may fill out anytime during or after the presentation. Finally, at the bottom pane are the icons to bring up all these widgets in case they are minimized or hidden. Again, if you are just joining us, I'm Brian Field, your moderator. Today we will be hearing from Professor Richard Van Bremen, and our topic is affinity selection mass spectrometry for the discovery of pharmacologically active natural products. All right, without further ado, let's get started. Richard B. Van Bremen is Professor of Pharmaceutical Sciences at the Linus Pauling Institute and the College of Pharmacy at Oregon State University. Professor Van Bremen received his BA in chemistry from Oberlin College in 1980 and his PhD in pharmacology and experimental therapeutics with Catherine Fenslow from the Johns Hopkins University in 1985. He carried out postdoctoral research in laser desorption mass spectrometry with Robert Cotter at Johns Hopkins before joining North Carolina State University and then the University of Illinois College of Pharmacy. At Illinois, he co-founded the UIC-NIH Center for Botanical Dietary Supplements Research with the late Norman Farnsworth and served as its director until moving to Oregon State University in 2018. He is on the editorial boards of Assay and Drug Development Technologies, Biomedical Chromatography, and Journal of AOAC International. His research concerns the discovery and development of natural products as chemoprevention agents and the investigation of botanical dietary supplements as alternatives to hormone therapy for menopausal women. And now I will hand it over to Professor Richard Van Bremen.
Thank you, Brian, for that introduction, and thanks to everyone at Shimadzu Scientific Instruments and Shimadzu Global for helping to make this webinar possible. Our laboratory is located on the campus of Oregon State University, where I'm Professor of Pharmaceutical Sciences in the College of Pharmacy. And my laboratory in particular is located in the Linus Pauling Institute, which was founded by two-time Nobel Prize winner Linus Pauling. And the Institute remains focused on the study of natural products, vitamins, minerals, and how their optimum dosages may help keep people healthy longer and prevent diseases of aging. Today I'm going to be discussing how we use affinity selection mass spectrometry to discover new drugs and compounds with pharmacological activity from natural product sources. But first, let me give you some background and history of how affinity selection mass spectrometry was discovered and what it was originally used to do. So originally, ASMS was invented to address the need for higher throughput screens and assays to discover compounds that had been synthesized as combinatorial libraries through combinatorial chemistry. Three ASMS techniques have become most successful over the years. They're called size exclusion LCMS, pulsed ultrafiltration or PUF MS, and magnetic microbead affinity selection screening. All of these techniques have in common the use of high-resolution accurate mass spectrometry connected to HPLC or UHPLC. And they all have their roots in screening mixtures of compounds, originally combinatorial libraries. In 1996, a group led by Gavin Dollinger applied for a patent and then published in the next year an approach called size exclusion LCMS. At that same time, my group invented pulsed ultrafiltration, or PUF MS, to do exactly the same thing, but through a different approach, all related to affinity selection and mass spectrometry. And a few years later, my group also invented the MagMASS approach, which differs from the other two only in that the target or receptor molecule is immobilized. Pulsed ultrafiltration and size exclusion affinity selection and mass spectrometry use a solution phase receptor or target for screening. All of these compounds are perfect for screening mixtures, whether these mixtures are equimolar combinatorial libraries or completely unknown natural product mixtures. They differ only in the approach used to separate the bound compounds from the unbound compounds after they're incubated with the mixture of compounds and the receptor. At equilibrium, size exclusion can be used to separate the unbound compounds from the receptor ligand complexes. Ultrafiltration may be, or a magnetic field may be used to separate the compounds bound to a receptor immobilized on a magnetic microbead. All ASMS approaches use high-resolution mass spectrometry interfaced with HPLC or UHPLC systems. In its original form, these approaches used HPLC, and more recently, my group and others have migrated to UHPLC because it's up to 8 times faster in the separation phase, and therefore one can screen more compounds, more libraries, more natural product mixtures per day. We use the Shimadzu UHPLC system and for the HPLC or UHPLC separations. And then we use electrospray or APCI on a high-resolution accurate mass quadrupole time of flight. We prefer the Shimadzu LCMS-9030 for this application. Whether one uses electrospray or APCI is a choice based on the types of compounds that one wishes to measure. Typically, the more nonpolar compounds are most amenable to APCI, whereas the more polar compounds work best in electrospray. To solve this dilemma about which to use, Shimadzu also offers a Duos ion source that combines both electrospray and APCI, and we also use this approach. High-resolution mass spectrometry is typically used online with LCMS to profile the compounds that bind after affinity selection. The accurate mass measurement is ideal for the determination of their elemental compositions. And then that information may be used to identify the natural products in a complex mixture or the combinatorial compounds from a library in a mixture. And to further differentiate compounds that might have identical compositions and masses, tandem mass spectrometry may be used as well. The invention of pulsed ultrafiltration affinity selection mass spectrometry has its roots in a collaboration I had at the University of Illinois at Chicago with colleagues Dwayne Venton and Chuck Woodbury. They were interested in monitoring and measuring binding of compounds as they passed through a stirred chamber containing a known macromolecule of a fixed concentration. An injection of a known amount of a potential ligand, a pulse, into a slowly flowing HPLC line into the stirred chamber could be monitored by the UV detector at that time, or I realized and added to the idea monitoring by mass spectrometry. The idea is that if the flow is slow enough, a near equilibrium condition may be reached inside this chamber such that if you monitor what is flowing through the ultrafiltration membranes out of the system to the mass spectrometer detector, that is the free concentration. Since you know how much was injected, you can calculate the bound amount. And then at multiple time points of monitoring, one can construct curves like binding enthalpy curves and so on, and determine all of these information constants about the binding. One can rank mixtures of compounds according to the affinity for the receptor. One can measure affinity constants, determine the stoichiometry of binding, enthalpy and entropy may be calculated. The entire binding kinetics profile may be determined. But of course, we realize that one could also screen mixtures of compounds and discover those that had the highest affinity for the particular macromolecule in the system. Here's an example of how pulse ultrafiltration may be used to measure the binding constants for a particular ligand, in this case warfarin, injected as a pulse into a stirred chamber containing human serum albumin as the receptor. And as the free concentration of warfarin is monitored by UV, mass spectrometry also works, of course, and if there's no chromophore, UV won't work, and that's why mass spectrometry is always going to be effective for this. We see concentration rise and then gradually decline down to baseline. This process is repeated without the receptor, human serum albumin in the chamber, and we see another curve, concentration over time. And what happens if there's binding between the injected ligand and the receptor is this curve gets spread out over time. The peak maximum is reduced. The length of time it takes to reach baseline is elongated. And we can calculate the bound from the free ligands at all time points. And then this information may be plotted as shown on the right-hand side of this slide. And in this case, warfarin has two distinct binding sites on human serum albumin, and we can calculate from the slopes of these lines the binding affinities. One is on the order of 10 to the 4th per molar, and the other on the order of 10 to the 5th per molar. Out of those early studies with pulse ultrafiltration affinity constant studies came the invention of pulse ultrafiltration affinity selection mass spectrometry as a screening tool to discover ligands to complicated mixtures of compounds, combinatorial libraries. And we were the first to also recognize that the libraries could be natural product mixtures, such as botanical extracts or extracts of microbial fermentation, broths, and so on. All of the ASMS approaches begin with the incubation of the mixture of potential ligands with the receptor of interest. And this receptor can be a protein, RNA, cell organelle, all kinds of large and molecules or combinations of them. After equilibrium is achieved, the bound compounds still retained by their receptors are separated from the unbound low molecular weight compounds by passing the low molecular weight compounds through an ultrafiltration membrane. Then the ligands on the receptor are released by a denaturing solution, which could be an organic solvent or a reduced pH binding solution. The released ligands pass through the ultrafiltration membrane. They can be trapped on a reversed phase column, and then LCMS is carried out. A control experiment may be carried out with denatured receptor, or even no receptor at all. And if ligands appear in the LCMS profile, that's due to nonspecific binding of to the ultrafiltration membrane, or perhaps to the denatured protein. And if we see an increase in peak area relative to the control in the experiment, that means the experiment has a ligand that is binding to the receptor molecule. And what's interesting about, and very important about ASMS screening, we can detect compounds that bind to the active site, orthosteric binding, as well as compounds that bind elsewhere, either cooperatively or allosterically. As an early application of affinity selection mass spectrometry to the discovery of natural products and other compounds that bind to important pharmacological targets, we developed an assay to find ligands to the retinoid X receptor, or RXR. RXR is one of 48 human nuclear receptors that includes retinoic acid receptor, PPAR gamma, vitamin D receptor, estrogen receptor, progesterone receptor, and so on. RXR dimerizes after it binds a ligand. It can also heterodimerize with about a third of these other human nuclear receptors. The function of these receptors, once they bind ligand, is to, in the nucleus of the cell, regulate DNA transcription. They can either downregulate the expression of genes or upregulate their expression. And RXR in particular is a very good target for cancer chemoprevention because it can stimulate cells, cancer cells in particular, to undergo apoptosis. It can prevent differentiation, or it can stimulate differentiation of normal cells so that they do not become cancerous. And it can downregulate growth factors, oncogenes, and tumor promoters, which could contribute to the development of tumors. And also important is there are relatively few RXR ligands known. We developed an ASMS assay based on pulsed ultrafiltration for the discovery of ligands to the RXR alpha human nuclear receptor, and some of the data are shown here. We used a positive control to develop and validate the assay, in which case this compound LG100268 is shown. And what we see is an LCMS chromatogram with a mass spectrometer response on the y-axis and the retention time on the x-axis. This was carried out using electrospray. And there are two traces, one solid line, a large peak, and then under that a small dotted line peak. The dotted line is the negative control, which controls for nonspecific binding to the apparatus. But we see peak enrichment due to the compound LG100268, which shows that it has bound specifically to the RXR alpha receptor. We then screened the negative control, the all-trans retinoic acid in particular, because the known receptor of RXR is a cis retinoic acid. So the all-trans form should not bind, and here you see two overlapping peaks, no peak enrichment, no specific binding, and therefore the negative controls are just what we expected for the all-trans form of retinoic acid. We screened a number of natural products and natural product analogs and found the compound AM636, which is a synthetic analog of the natural product camptothecin, which is a topoisomerase inhibitor. And this compound AM636 was synthesized by our colleague Mark Cushman at Purdue University in a collaboration. And what we found was that AM636 bound specifically, as you can see, based on the peak enrichment relative to the control. We can also test for where the compound bonds to the receptor by adding a large amount of a known ligand of high affinity, such as the LG100268, and by adding the competitive compound at high concentration, the AM636 should not bind anymore if it is competing for the active site, the orthosteric site, as does the LG compound. And that's how we confirmed that the AM636 bounds to the same site, the orthosteric site, as did the LG positive control. Had these two compounds not competed for the same site, that would show that the AM636 compound was binding to a separate place, and it would be an allosteric ligand. A number of other analogs of camptothecin, all indeno isoquinolines, were screened using this PUF MS approach. Equimolar compounds were screened, and then the relative binding to the RXR receptor could be rank ordered by the peak area of each of these compounds as detected by LCMS after affinity selection pulsed ultrafiltration. And note that the order of ranking from very high to very low is identical to that which we determined in a functional assay using an RXR transcriptional induction approach. Therefore, in a single step, we can test a mixture of ligands, rank order them by affinity for the active site of the receptor, and this order is mirroring exactly what happens in vivo. As a rexinoid, that is a ligand that binds to the RXR receptor. We wanted to make certain that AM636 did not also bind to the retinoic acid receptor, and that assay using RAR gamma showed that AM636 was selective for the RXR and not the RAR. Additional biological tests showed that AM636 as a cancer prevention agent could inhibit the growth of the MCF7 breast cancer cell line. It produced G2M phase arrest at relatively low concentrations. It could also enhance S phase arrest at higher concentrations. It upregulated the expression of target genes of RXR, showing that it did target the RXR in cell culture. And it induced P21, or it prevented the induction of P21, which is important in cancer prevention as well. And in animal in vivo models with rats, AM636 attenuated the growth of induced mammary tumors induced by the carcinogen N-methyl N-nitrosourea. Like RXR, the vitamin D receptor is a member of the nuclear receptor superfamily. And the classic role of vitamin D receptor ligands like calcitriol is regulation of calcium and phosphorus in bone metabolism. But we wanted to test AM636 and other compounds to see if they were also ligands to the vitamin D receptor, which is also of interest as a cancer chemoprevention target. And ideally, a vitamin D receptor, which could function as a chemoprevention agent, should lack the bone activities of vitamin D receptor ligands. The new vitamin D receptor PUF MS assay was used first to test curcumin, a known ligand of the vitamin D receptor, for validation purposes. And as you can see in the red trace of signal detected versus time in the PUF ASMS chromatogram, curcumin binds very tightly to the vitamin D receptor relative to the control, the dotted line. The internal standard genistein, here shown in blue, was added just before the LCMS analysis, so that the experimental in the solid line and the control in the dotted line could be normalized to show that curcumin indeed was enhanced due to specific binding to the active site of the vitamin D receptor. Then we screened AM636 to see if it were also a ligand to the vitamin D receptor, as it had been for the RXR. And in green, you see the enhanced peak relative to the baseline, which is basically zero. And this peak enhancement shows that AM636 is also a ligand for the vitamin D receptor. The amounts of the receptor and the test compounds are shown here, and they're in the low micromolar range. Note that the micromolar versus nanomolar activity is something you, the operator, selects in advance. If you wish to detect compounds in the micromolar KD range or better, choose a 1 or so micromolar receptor concentration. And then one can detect compounds in the micromolar KD range or higher affinity. If you wish to move the bar down and only detect compounds with nanomolar affinity or better, then reduce the receptor concentration to the nanomolar level. Although my laboratory typically uses PUF, ASMS, or magnetic bead affinity selection screening, I wanted to show you briefly how the size exclusion chromatography ASMS approach works. Again, like PUF MS, it begins with the incubation of the ligand mixture with a solution phase receptor. After that comes to equilibrium, the bound compounds on the receptors are separated from the unbound low molecular weight compounds by size exclusion chromatography. In size exclusion, the large molecules are leave first, that is the ligands bound to the receptors. Then the fraction containing the receptors is then loaded onto a reversed phase column, denatured with solvent like acetonitrile, and the ligands are eluted and you can see them elute in the chromatogram. And so from this point on, the reverse phase LCMS approach is identical to that of POF MS. Researchers at Pfizer and Merck have automated this approach with 2D chromatography such that the size exclusion and the reverse phase steps are carried out online with mass spectrometry detection. And they've typically been screening pooled library compounds, typically about 2,000 compounds each, and they can do about 100,000 compounds a day this way. Because the combinatorial library compounds have known compounds, one can program a computer database so that once these compounds are measured by mass spectrometry, you can deconvolute and determine what the hits are in each sample. Folks at Merck, Rizvi and Nickbar, have shown that RNA can also be used as an alternative to protein targets. So the macromolecule can be any macromolecule. Our invention, magnetic microbead affinity selection screening, differs from the other affinity selection mass spectrometry approaches in that the receptor is immobilized on a magnetic microbead. AF-MS techniques of POF and size exclusion use solution phase ligands only. So this is important because some ligands are not water soluble. For example, those that are embedded in membranes in the endoplasmic reticulum on the cell surface or on the membranes of the mitochondria or nucleus, for example. So after the compound of the receptor is immobilized on the magnetic bead, the process is the same as the other AF-MS approaches. Those beads containing immobilized receptors are incubated with the compound mixture, which could be a combinatorial library or a natural products mixture. After equilibrium, the bound from the free are separated using a magnetic field. This approach is readily amenable to microtighter well plates, like a 96 well plate approach, where a large magnet under the entire plate can hold the beads in place while pipetting removes the unbound compounds. Replacing the binding buffer with a denaturing solution while the beads are still held in place can release the ligands, which can then be taken out of the well and injected onto the LCMS system for LCMS analysis by accurate mass high-resolution mass spectrometry and tandem mass spectrometry. A quick note about the use of our MagMASS approach. Quartararo and others at MIT recently reported the use of this approach to screen ultra-large peptide combinatorial libraries, which contained up to 10 to the 13 members. They found that they could detect ligands to an antibody receptor in libraries with as many as 1 million, even up to 10 to the 8 compounds per single well of the experiment. So this work is important because it demonstrates the upper limit of the complexity of the combinatorial library, and I suppose that could extend to the natural product mixture of maybe being useful for up to 1 million compounds at once. Now that we've discussed each of the AF-MS approaches that are used most today, POF MS, size exclusion, and MagMASS, this chart is a comparison of the different techniques. And I'll just show you briefly how they are alike and how they are different. They are all suitable for the screening of combinatorial libraries with about 2,000 compounds per mixture in POF MS or size exclusion ASMS. At least 1 million, maybe even more compounds in combinatorial libraries can be screened using MagMASS thus far. All are useful for the screening of natural products and mixtures. All of them use less reagent to screen a large number of compounds than would, say, conventional high-throughput screening approaches. AF-MS requires no radiolabeled compounds, no chromophores, no fluorescent tags, no fluorescent reagents. It's compatible with all kinds of compounds, whereas fluorescent compounds often interfere with conventional high-throughput screening. Mass spectrometry is blind to that, and such chromophores do not interfere. ASMS is suitable for any type of assay buffer, any type of cofactor. Even if the target is understudied, relatively new, and you don't even know what the affinity site is, one can still use ASMS to screen for ligands to it. ASMS, of course, finds orthostatic ligands that bind to the active site, but unlike conventional high-throughput screening, ASMS also finds allosteric ligands. We can rank mixtures of compounds by affinity to a receptor in a single experiment, which would take multiple experiments otherwise. We can measure affinity constants by PuF MS, and it's possible using other approaches, but we've done a lot more work with the PuF MS applications of affinity constant measurement. PuF and size exclusion are, as I mentioned before, designed to look for ligands to solution phase targets. MagMASS requires immobilization, so that makes MagMASS ideal for looking for ligands to solid phase or immobilized receptors. PuF MS and the magnetic bead approach allow reuse of the receptor. The size exclusion approach does not. Now, to compare affinity selection mass spectrometry with conventional high-throughput screening, both approaches work fine for combinatorial library screening, with at least 1 million compounds at a time possible by ASMS, whereas conventional HTS looks at 1 compound at a time. Consumption of the reagents, if you're looking at multiple compounds at once, is much lower in ASMS, much lower using ASMS compared to HTS. ASMS is better for natural product mixture screening because natural products are often rich in fluorescent compounds that can interfere with many HTS approaches, such as fluorescence polarization screening. No labeled compounds, reagents, no chromophores, or no fluorophores are required for ASMS, but they are typically used in conventional HTS. It doesn't matter what the buffer is, what the cofactor is, what the target is, what the ligand is. ASMS works with everything, but this is not the case with conventional high-throughput screening. We can measure affinity constants with ASMS, not so easy using conventional high-throughput screening, at least not in a single experiment. We can look at solution phase or solid phase in both approaches, whether the receptors are mobilized or in solution. And the ASMS approaches with PuF and MagMASS allow reuse of the receptor, whereas conventional high-throughput screening does not. Natural products have always been a source of drug discovery. Even today, more than half of all therapeutic agents are derived from natural products. More than 2/3 of cancer drugs are natural products or inspired by natural products. This schematic illustrates how most natural products chemists today discover new compounds that have pharmacological activity. They make an extract of a botanical or a natural product from a marine organism or a bacterium or a fungal extract and test the extracts in various bioassays. If activity is determined, the extract is simplified through fractionation, maybe by column chromatography. Each fraction that is collected is retested in the bioassay. The active extract is refractionated, subfractionated in a repetitive iterative process until an active compound is isolated, and then it can be characterized using mass spectrometry, NMR, and so on to determine the chemical structure of the active constituent. And of course, that active constituent would then be retested in the bioassay for activity. ASMS enables this active extract to be characterized immediately. And this process is therefore incredibly fast relative to the conventional approach. Literally, we can do in a few hours what it takes days to weeks to do by the bioassay-guided fractionation approach. Cyclooxygenase 1 and 2 are important enzymes that convert arachidonic acid to prostaglandins and related compounds that are inducers of inflammation. Anti-inflammatory agents such as the non-steroidal anti-inflammatory drugs target COX-1 and COX-2. Aspirin and ibuprofen, for example, inhibit both of these enzymes, whereas the more recent drug celecoxib is selective for inhibition of cyclooxygenase 2. COX-2 is the inducible form that it becomes most problematic during times of inflammation and is important in the symptoms of inflammation, swelling, pain, and so forth. So anti-inflammatory agents that inhibit COX-2 are particularly important to help treat inflammatory problems. And so we developed an ASMS assay for COX-2. To validate our new assay for cyclooxygenase 2, we used a known synthetic ligand for it, an inhibitor, celecoxib. The LCMS assay after the pulsed ultrafiltration affinity selection is shown here. Here the dotted line is the celecoxib peak relative to the control, which was the celecoxib and buffer without the enzyme. And we see, as expected, here the retention time of about 15 minutes during the LCMS separation. The celecoxib peak is greatly enhanced relative to the negative control. And this was monitored using LCMS, positive ion mode, and the celecoxib ion is detected at mass to charge 382 in positive ion electrospray. As a natural product application of our new COX-2 QToF MS assay, we screened an extract of ginger root for compounds that could inhibit COX-2. Historically, it's been known that ginger and preparations of ginger have anti-inflammatory properties. So we wanted to find out if any of these compounds are actual inhibitors of COX-2 as well. And what we found in our assay was that components of the gingerol group, like 10-gingerol, chogols like 8-chogool, pyridols, and even some ginger diones and dehydrogingerols are inhibitors of COX-2. We also have a parallel assay to look for inhibition of COX-1. Of course, these are all binding assays, and once we identified an active compound this way, we then moved to a functional assay to show that they were indeed inhibitors, and we could compute IC50 values to determine the concentration required to inhibit 50% of enzyme activity. And those assays showed that gingerols and chogols and pyridols are typically in the low micromolar IC50 range. Another chemoprevention target that we developed an assay based on QToF ASMS for is Quintin Reductase II. In this particular chromatogram, we detected a QR2 ligand in a marine sediment bacterial extract. The marine bacterium was cultured by our colleague and collaborator Bill Fenical at Scripps UC San Diego. This particular ligand, relative to the dotted line background, was a high affinity QR2 ligand with an accurate mass measurement of 292.0600. Now, because we have the accurate mass measurement, we can compute the elemental composition and then compare that to databases of known natural products as well. And this is the beginning of the process to identify what the compound is. Also, we confirmed this compound bound specifically to the active site of QR2 through competition with some known ligands. The next step is to compare this compound with spectra of known compounds for identification. So based on HPLC retention time, accurate mass measurement, and tandem mass spectrometry, this compound from our QToF MS screening experiment, eluting at 8.4 minutes, was identical both in mass, retention time, and in its tandem mass spectrum to the known compound of amosamide B. So in this process, we could identify the compound quite quickly because it was already a known and characterized natural product. In addition to QToF ASMS, my laboratory uses MagMASS, microbead affinity selection screening quite extensively. And as a reminder, this approach begins with the incubation of an extract of natural products or a combinatorial library mixture with a receptor immobilized on a magnetic bead, a microbead, which can be retained in a 96 well plate to enable the separation of the bound from the free very quickly. One of the early applications of MagMASS in my laboratory was the screening of natural products and botanical natural products in particular for ligands to the estrogen receptors, ER alpha and ER beta. Shown here is the MagMASS the MagMASS LCMS profile of compounds from an extract of the hop plant. Hop cones contain compounds like xanthohumo, other prenulated flavonoids, and phenols. And we found one compound, if you look at the red trace relative to the dotted background line, we see enhancement for the 8-prenylnaringenin. I'm showing all of these compounds because they are structurally similar and at relatively high concentration in hops and hop extracts. But only the 8-prenylnaringenin peak showed lots of enhancement for binding to the estrogen receptor beta. Here are the chemical structures of the hop compounds with the prenyl group as I showed you before. This is xanthohumo. It can cyclize to isoxanthohumo, which has very weak estrogenicity. But it can be OD methylated with the removal of a methyl group from a phenolic position, either by gut bacteria or by the human cytochrome P450-1A2 to form 8-prenylnaringenin, which is one of the most potent phytoestrogens that has been yet identified. And this is the compound identified in our hop extract by MagMASS. Magnetic microbeads for MagMASS come in a variety of surface chemistries, which facilitate the covalent immobilization or the non-covalent immobilization of the receptor molecules to the beads. In some of our work using retinoic acid, retinoid X receptor alpha, we've immobilized the protein through nucleophilic amino groups to the magnetic beads activated with N-hydroxyl succinimide, or NHS, on the surface. This forms a covalent bond, which facilitates the reuse of the enzyme or protein, which is a receptor in this case, and also helps stabilize it for screening purposes. Alternative covalent immobilization approaches are available using either surface chemistry of the beads activated with carboxylic acids or amino groups, and then a carbodiimide could be added to activate the immobilization of the protein, either an amino group of the protein to the carboxylic acid or the carboxylic acid of a protein to the amino group on the bead. In addition, sulfo-NHS chemistry is available as a covalent activated surface. Non-covalent immobilization is also possible, and we often use this approach if the target macromolecule has a biotin tag. One can immobilize it on added and derivatized beads. His tag proteins are readily immobilizable on an NTA nickel surface chemistry. And a maltose binding protein, if it's co-expressed with the protein target of interest, can be immobilized on an amylose derivatized bead. Other surface chemistries are available as well. Always one of the goals of high-throughput screening is to automate the system as much as possible to eliminate or reduce the hands-on manipulation. And one of the goals, of course, is to try to use and implement multi-titer well plates. Multi-well plate formats have been most successful in the MagMASS approach. Limited success has been realized in the puff MS approach because 96 well plates containing an entire sheet of ultrafiltration membrane are available. But the MagMASS approach really works well, and it facilitates the automation of MagMASS, and it's one of the most successful automated approaches yet in ASMS technology. To complement the cyclooxygenase anti-inflammatory target assays, we developed an assay for ligands and inhibitors of 15-lipoxygenase-1, or 15-LOX1 for short. 15-LOX1 converts arachidonic acid into inflammatory compounds such as 15-HP and others. It converts linoleic acid into an inflammatory compound, as well as has an interesting effect with docosahexaenoic acid in that this product actually, and its downstream products, reduce inflammation. So if arachidonic acid and linoleic acid, which are very abundant in the human body, are substrates, the products are inflammatory and promote asthma, oxidative stress, and other inflammation-mediated problems, and contribute to the inflammation of atherosclerosis. On the other side, on the other hand, DHA, if it's available in human body through dietary means, but DHA is converted to beneficial compounds. But in having the goal of having inhibitors of 15-LOX actually has potential for the prevention of heart disease as well as some neurological diseases. To validate our new assay for 15-LOX1 using MagMASS, we used the compounds gluteolin, which is a known ligand with a 5 micromolar Kd, and a non-ligand, epsilon. After incubating gluteolin and epsilon at equal molar concentrations with 15-LOX, here is the LCMS chromatogram after a MagMASS separation. We see peak enrichment for luteolin, but not for epsilon. So our positive controls showed affinity for 15-LOX1 as expected, and epsilon did not as expected. So this validated our assay, and then we turned our attention to screening natural products for new ligands to 15-LOX1. We applied our new MagMASS assay for the discovery of ligands to 15-lipoxygenase-1 to the testing of extracts of 14 North American prairie plants used historically by Native Americans to treat inflammation. In this case, the data that you see here were obtained using a high-resolution Shimadzu IT time-of-flight mass spectrometer, a forerunner to their current QToF instrument. Polarity switching was used, so above the line and below the line are negative and positive ion chromatograms. And the data were processed online using XCMS metabolomic software. And this helped us compare the experiment to the control chromatograms and identify peaks that had been changed due to peak enhancement as a result of selective binding to 15-lipoxygenase. Only one of the 14 prairie plants, the marsh mermaid weed, had a positive result, and this was the peak here at about 8-minute retention time, which showed enrichment due to specific binding to 15-LOX. Affinity selection mass spectrometry with a high-resolution mass spectrometer gives us elemental composition information, which we can search online against natural products databases, such as you see here, like MassBank, or the Scripps Center for Metabolomics, Metlin, MOVZ Cloud, Reaxys, SciFinder, and GNPS. So we search these databases and identify the compound, actually two compounds with the right elemental composition for our lead compound in our lipoxygenase assay. Two compounds came up in our natural products database searching based on elemental composition as potential ligands and inhibitors of 15-lipoxygenase. One was quercitrin, the other was 7-luteolin glycoside. And both are available commercially. We obtained them and tested them in our assays, and quercitrin was positive as an inhibitor of 15-lipoxygenase-1. In the literature, it turns out quercitrin is actually already known to be an inhibitor of 15-LOX1 with an IC50 value of about 80 micromolar. In our continuing effort to automate and increase the productivity of MagMASS, we recently applied the Kingfisher Flex Magnetic Particle Processor to MagMASS. Although this instrument was originally developed for RNA and DNA purification, we found it could enhance the throughput of MagMASS about threefold and increase the sensitivity of the screening about fourfold. And this is because of the automation and speed. The process, of course, begins with the immobilization of the receptor to the magnetic particle, which is then incubated with the natural product or combinatorial library mixture. At this time, instead of retaining the beads in the bottom of the 96-well plate well with a large magnet, a magnetic probe is inserted into the well to remove the beads, which can then be washed and then moved to a denaturing solution to release the ligands. And then those ligands can be injected into the LCMS for analysis. So this process is more sensitive and faster than our previous MagMASS approach. We want to thank Brett Tyler and his team at the Center for Genomic Research and Biocomputing at Oregon State for making this instrument available and helping us develop this assay. This is a side-by-side comparison of our previous MagMASS approach compared to the Kingfisher-assisted approach. And as I said before, it begins with the incubation, but instead of retaining all of the beads on the bottom of the well with a large magnet and then washing with pipettes, the probe simply captures the beads electromagnetically and moves them from well to well for washing, release of the ligand, and then we can inject the released compounds into the mass spectrometer. And for my last application of affinity selection mass spectrometry, I'd like to tell you the progress we've made thus far in screening for compounds that can inhibit the SARS coronavirus 2 responsible for COVID-19. The SARS virus infects human cells by first binding to a receptor on the human cell surface called ACE2. And the spike protein of the virus contains an amino acid sequence which recognizes the ACE2 protein. And this is the process by which the virus begins to attach and then invade the human cell. And so that if one could block this interaction between the spike protein and the ACE2 of the human cell, one could block and prevent infection of the human cell altogether. So we developed an assay for ligands to the spike protein surface. And in particular, the spike protein is a trimer. And one could either use the trimeric oligomer for screening or the monomer for screening. And I'll show you data based on the S1 subunit monomer, which was synthesized recombinantly using a His tag. We immobilized the His tagged spike protein monomer from the SARS coronavirus 2 on magnetic microbeads derivatized with a divalent nickel on their surface. We validated this new assay using the SBP1 peptide, which is the recognition amino acid sequence of the ACE2 human protein, which is on the surface of the human cell. So the SBP1 peptide should bind to the spike protein. And as you can see with the peak enrichment in the black line versus the nonspecific binding control, red line, this peak enhancement shows that the SBP1 peptide does bind to the spike protein in our MagMASS assay. We are using our new MagMASS assay to screen natural products from a variety of sources for ligands to the SARS coronavirus 2 spike protein. This work is ongoing and is using electrospray on the Shimadzu LCMS-9030 QToF mass spectrometer. Shown here is a UHPLC-MS chromatogram of an extract from a single botanical source that contains at least 3 ligands to the SARS coronavirus. Work is ongoing to determine if these ligands can inhibit the spike protein from infecting human cells, and I hope to be able to share the results of that work in the near future. So what's next for affinity selection mass spectrometry? For natural products applications, work is continuing to develop a comprehensive natural product database that can combine a variety of existing databases into something that's searchable by mass and elemental composition. Existing metabolomic software programs from a variety of sources can be adapted to become more user-friendly for ASMS. And of course, work is continuing to automate and increase the throughput of all the affinity selection mass spectrometry techniques, in many cases implementing multi-titer well-plate format. New targets are being adapted to ASMS screening all the time, and these targets are including entire cell organelles like mitochondria and membrane-bound targets as well. In addition, work is continuing with ASMS to discover allosteric ligands as well as orthostatic ligands. This is a particularly exciting area of discovery for which conventional high-throughput screening is just not as amenable as ASMS. And of course, we're doing our best to continue to automate natural products drug discovery. Finally, I'd like to thank all of you for tuning into this webinar and for your attention. I want to thank the sponsors at the NIH, of course, Shimadzu Scientific Instruments for making this webinar possible. I want to thank Youngmei Lee at Avon Bio for supplying the magnetic microbeads to be used. All my colleagues over the years at the University of Illinois at Chicago, especially Diana Kolich. I want to thank Bill Fenical at UC San Diego, Brett Tyler at Oregon State, Mark Cushman at Purdue, all the former group members who contributed to the invention and development of pulsed ultrafiltration, MagMASS, and other ASMS techniques. And of course, I want to thank my current group members, and in particular, Ruth Maturi, who's in the middle of this group photo, who has been so helpful in the development of MagMASS and applying it to the discovery of ligands for the coronavirus spike protein.
Let's thank Professor Richard Van Breeman for such an exciting presentation. Again, under the resource list, you can find more information about our analytical solutions. If you have not had a chance, please fill out the survey questions on the right-hand side to provide feedback and request additional information. We also encourage you and your colleagues to attend our upcoming webinars, which encompass a wide variety of application areas. The registration link is located at the bottom of this slide. At this time, we will begin our Q&A session. Thank you to the audience for attending and sending questions. We have time for just a few questions. Here's a good one. Among the three approaches, which one is the best to get rid of weak binders?
I think what the question is asked after is whether or not there's background noise or nonspecific binding. If the question is after how do you determine whether the binder is a high affinity, a medium affinity, or a low affinity ligand, that's another matter. But set the concentration of the receptor molecule at the concentration at which you wish to find the KD ligand. So a micromolar KD receptor will help you identify from 10 micromolar KD ligands on down to the micromolar, submicromolar, nanomolar, and so on levels. If you don't want to detect micromolar ligands, set the concentration of the receptor molecule lower. 1 nanomolar receptor will enable you to detect 1 to 10 nanomolar KD ligands and lower still like the femtomole KD level. If the idea is about how do you reduce or eliminate background noise, well, what counts is when you do the experiment to control for nonspecific binding, compare chromatograms of the experiment to the control with a denatured receptor or no receptor at all. And if there's nonspecific binding to the apparatus, like the ultrafiltration membrane, we look for enhancement of the peak during the affinity selection LCMS chromatographic separation. And if there's peak enhancement, that's specific binding. All the techniques have a little bit of nonspecific binding. Some are better than others. I think the size exclusion approach reduces the background noise probably more than the other approaches, but it has another problem in that it takes longer to separate the bound from the free than, say, the MagMASS approach. Speed is of the essence if you have a compound which dissociates quickly from the receptor. The longer it takes to separate the bound from the free and then do the final LCMS profile, the more likely you are to lose sensitivity due to dissociation of that ligand from the receptor. So in that sense, the MagMASS approach we found to be the fastest and the most sensitive. Next question, please.
Yeah, great, thank you. Along a similar line, they're wondering whether or not the automated process reduced the weak binders amount.
Well, certainly we found that any automated system that is faster than you can do it by hand enhances the sensitivity and productivity because it's faster. But faster, as I said, is important during that separation step. Once at equilibrium, of course, you have the maximum number of ligands bound to the receptors. Once you separate the unbound compounds from the bound ligands, dissociation and loss of ligand begins. So you want that process of getting from the incubation mixture to the mass spectrometer to be done as fast as possible.
Great, thank you. Yeah, so regarding fast polarity switching, do you run your ASMS with fast polarity switching with your natural products?
I showed an example which was carried out on the QToF mass spectrometer where we did polarity switching to find the natural product quercitrin. And that was a 15 lux ligand. So we typically, yes, run both positive and negative. One can run a positive LCMS profile and then a negative one as a separate run, or you can do polarity switching. Of course, the faster the polarity switching, the better if you have a very quick HPLC or UHPLC separation. So the rate of polarity switching is really determined by the peak width of the chromatography. If the chromatography is relatively slow, so that peak width is, say, 10, 12 seconds, one can do a 1 second polarity switching and get enough points across that peak in positive mode and then also in negative mode to define the peak. But if you're using UHPLC and the peak width is 20 seconds, I mean, not 20 seconds, it's 3 seconds or 4 seconds, you might not get enough points across the peak to determine it if you're using 1 second polarity switching. So in that sense, the faster the polarity switching, the better it. Make sure it's matched to the chromatography.
Great, thank you. Let's see. So here's a question. MagMASS appears to be very useful for a wide range of applications, such as drug discovery, biomarker discovery, combinatorial screening, and diagnostic assays. If there are attendees today that would like to apply MagMASS to their research, what is the best way to proceed? Will commercial MagMASS kits be available soon?
Is your question about the separation process or the beads themselves?
I think about the beads themselves. If somebody wanted to implement this technique in their labs, how would they go about doing that?
The magnetic microbeads have been available for some years and were originally developed and marketed to the RNA and DNA synthesis and sequencing and processing laboratories. Little by little, we're seeing them being repurposed for other means like affinity capture, protein precipitation, things like that. Our approach is to repurpose them, and in that we immobilize the receptor and use that for our affinity selection mass spectrometry. The beads are commercially available from several sources with different surface chemistries. And I described some of those chemistries in my slides. They could be covalent or non-covalent immobilization, surface chemistries with amino groups, carboxylic acid surface groups, and so forth. Or affinity tags. So you can have avid and biotin interaction, amylose, maltose interaction, and so on.
Great, thank you. We have time for just a couple more questions. If we don't get to your questions, we'll be happy to respond to those after the webinar by email. So you mentioned in your What is Next slide that you intend to improve upon a user-friendly metabolomics interface for ASMS. Can you please elaborate here on what specific capabilities or features you would like to build into this new interface?
Well, what we found over the last few years is that the ability to do online searching of natural product structures and masses is getting better all the time. We have our own internal natural products library, but there's many more available online. Some of the libraries I mentioned in one of my slides, like the GNPS, SciFinder, Metlin, and so on, we search them all when we have a hit in our natural products drug discovery program. I think we'll see a gradual unification of them so that one day we'll be able to go to one place and search multiple databases at the same time. At this time, not all of these have elemental composition. Not all of them have accurate mass measurement that we can, exact masses so that we can compare our exact mass measurements to the true values. But eventually we're going to get there.
Great. One final question. How do you determine which plants to study? Is there a set of criteria that you use to study one plant over another?
Well, we certainly follow the literature when determining where to start. So if we're studying compounds that are, let's say, anti-inflammatory compounds, and we have an anti-inflammation target like 15 LOX of Cyclooxygenases, we go to the botanical literature and see where the ethnobotanical history is. If there are certain plants that have a history of use for that purpose, like relieving fever, inflammation of a particular sort, that's where we'll begin. We'll obtain plants or other natural product source material according to the ethnobotanical history.
Okay, great. Well, I want to thank everybody for all of the great questions. There's a couple unanswered questions. We will reach out individually to each of you whose questions we were not able to answer today. Once again, thank you all for attending and participating. We will send you an email and a link to view a recorded version of the presentation anytime. Again, we encourage you and your colleagues to attend our upcoming webinars, and the registration link is located at the bottom of the slide. I hope you all have a great day, and I'd just like to thank Professor Richard Van Breemen again for his time in going through this really wonderful seminar.
Well, thank you, Brian, and thanks to everybody for tuning in. I'll be glad